Greetings everyone. So today we're going to discuss about the bacterial genome. So bacterial genome here is what we call as distinguished into two major regions. There are two major types of bacterial genome present in a bacterial cell. Number one is the main bacterial genome, or what we call as the chromosomal genome, which is known as the nucleoid. This genome, which is known as a nucleoid, basically it is found in what we call as a region of cytoplasm, which is known as a nucleoid region. Now, since bacteria do not have any sort of membrane bound organelle, no nucleus is present, no nuclear membrane is present, so they are having their nuclear material found inside the cytoplasm, and that region is known as nucleoid region. And that is a region which is bit dense region as compared to the rest of the cytoplasm. And in that region we are having a circular DNA. Unlike the linear DNA of eukaryotes, there is a circular DNA, but again that is a helical DNA. Helical, double stranded. It is a helical, double stranded, circular, helical, double stranded, circular, haploid chromosomal DNA is present. Haploid chromosomal DNA is present. Helical double-stranded circular haploid chromosomal DNA is present, and that chromosomal DNA is haploid, which means that there is only single set of chromosome rather than a double set of chromosome. As we are having humans, in humans we are having a double set of chromosome, which is 2n, which is equal to 46, right? But when it comes to the bacterial chromosome, there's no haploid uh, bacteria. There's no what do we call it, the diploid structure present. So diploid genome is not present, rather there is an haploid genome, and that haploid genome is a single set of chromosome, which is known as the haploid genome, due to which these are known as haploid chromosomes. Haploid chromosomes, and these are known as the haploid organisms. So bacteria can be considered as haploid organism because they have a single set of chromosome. After that, the nuclear region is responsible for controlling the growth and metabolism of the bacteria. Growth and metabolism of bacteria. Growth and metabolism of bacteria. Right. Growth and metabolism of bacteria. While when it comes to the plasmid basically involves. It is basically also uh, the genetic material, but that is something we call as the extra chromosomal DNA. It is an extra chromosomal DNA rather than the chromosomal DNA. It is an extra chromosomal DNA. Again, it is circular. It is circular, as well as uh, it is double stranded as well, circular as well. It is extra chromosomal, which means it is not involved in the major chromosomal activities which are done by the nucleoid, but rather it is extra chromosomal and is not involved or associated with the growth and metabolism or the daily life activities. Growth and metabolism, but rather it is associated with unfavorable conditions. As the nuclear region is associated with favorable conditions, here it is associated with the unfavorable conditions, such as any sort of attack by chemical changes or chemical drugs or such stuff. Example of this includes ampicillin. If we um, in, ingest ampicillin, and then bacteria is exposed to that ampicillin present within the body, then the plasmid basically has a capability to resist uh, ampicillin, because it contains ampicillin resistant genes. Ampicillin resistant genes, which are known as PS, found in the region which is known as PSTI. PSTI. Now it is not same for all bacteria. Some also have these genes and some do not have. It's just an example of a particular bacterium, and it's not same for the each and every bacteria. So here, likewise, we are having tetracycline is the drug. So there are some bacteria which have tetracycline resistant genes present, due to which BAMHI given in the diagram as well. And these genes basically help to improve the immunity of the bacteria ultimately, due to which these are responsible for improving the bacterial immunity. Apart from which, when it comes to some other characteristics, Plasmid is also utilized in recombinant DNA technology. Recombinant DNA technology, which means that recombinant DNA is basically a type of DNA which is formed by the manipulation of a certain number of genes of a particular DNA and replacing them with the gene of interest. Let's suppose if we take up the example of um, the use of recombinant DNA in biotechnology, such that in biotechnology we utilize a terminology which is known as gene of interest. Let's suppose if we want to synthesize insulin. So in order to synthesize insulin, we need a particular organism's gene. We need the particular gene which is responsible for the synthesis of insulin within an organism. So that gene which is of our interest and can provide us our desired product, which is insulin, is known as a gene of interest. Gene of interest, right? And what we are going to do is to take the gene of interest, and then we are going to insert that into our organism. Insert that into an organism. Let's suppose we are taking bacteria which is having a plasmid. So let's suppose we are taking E. coli. So E. coli having plasmid, so we are inserting the gene of interest into the plasmid. How we are going to do is it actually is in such a manner that we are going to take the plasmid, and now we are going to utilize an enzyme, which a restriction enzyme is utilized in this condition. A restriction enzyme will be utilized, and that restriction enzyme utilized will be known as restriction endonuclease. That would not be endonuclease, because endonuclease is associated with the destroyal of the entire bacterial genome or entire genomic material. But when it comes to restriction endonuclease, it is only having its activity which is to a restricted extent. It means that it cannot completely destroy the bacterial genome, but rather it would be just responsible for the cutting of the bacterial genome and restricting some of its region, due to which this is known as restriction enzyme, known as restriction endonuclease. And apart from this, when we are going to utilize this enzyme, which is restriction endonuclease, that will cause our, particular portion, a particular portion of the a particular portion of the bacterial plasmid to cut off from here, due to which we are then able to insert our gene of interest. Let's suppose, let me just highlight it with green color. Gene of interest would be inserted over here. Gene of interest, right? Gene of interest. And once it is done, um, again we are going to utilize another enzyme for, what do we call it, attaching it. We have ligase enzyme for it. Ligase is basically, ligase particularly we can give it ligase enzyme is synthetase, but particularly we are having a name for it which is known as DNA polymerase. So we are going to utilize that enzyme, and in order to attach that, we are going to, attach the gene of interest. And once the gene of interest is attached, now here, this organism, uh, which is having that plasmid, has now become a genetically modified organism. E. coli, which was previously, E. coli, now it is basically considered as GMO, genetically modified organism, because we have altered its genomic sequence. Now, uh, the gene of interest is carried in the plasmid, so we can call plasmid as the carrier of the gene, carrier of gene, right? And after that, when we are going to leave it, for some time, after a couple of time, we are going to utilize that plasmid to synthesize our insulin, and of course we'll get our desired product. Now when it comes to the number of genes in plasmid, there are around 5 to 100 genes in plasmid, while when it comes to the nucleoid, there are approximately 3,500 genes. There are approximately 3,000 genes. E. coli is an exceptional case which has around 5,000 genes, and it has alteration in at least one of its gene in out of 200 species of the E. coli bacteria, while 5,000 genes are found. Moreover, when it comes to the nuclear, it is having 3,500 genes. That's all about the video. Hopefully we meet again in another video.